Hello. Excuse me. We got to start the new session. And this is another exciting session with interesting topics, and I will hand the microphone to my friend Tuman Wology. Tuman will chair this session and will make sure that everything is going according to time. So there is time limitations here. Please, Tuman.
Good afternoon. I believe everybody highly energized for this afternoon sessions after this nice toast. This afternoon we have 4 sessions, 4 presenters, and first one is titled Genome-Wide Association Studies of Body Morphometrics, Biomechanics, and Behavior in the dormitories, and presented by Dr. Elena Ciani.
Okay, thank you. So, again, this presentation was supposed to be given by Carlos Pastrana, who could not attend, so I will be given at his place. This time I'm going to talk to you about this phenotype-genotype association study on the traits that are mentioned in the title. Just to mention that these traits, so zoometric, biomechanics, and behavior, they can be considered as a sideline traits compared to camel milk and camel meats, but please put attention on how they can be relevant not only for milk and meat production, but also in many other activities where camels can be used for, like if we consider beauty contests or athletic, like racing and riding, or if you consider the use of the assisted interventions of camel with special people, and obviously the routine husbandry practices as what concern behavior. So we do think that zoometrics, but also biomechanics and behavior traits are also relevant and need to be considered and improved. It's fine? From this way? Okay, thank you. I can use this one otherwise. Okay, thanks. Okay, so the objective was to do this genome-wide association study. Is a small study for a genome-wide association study. We had 120 animals. They belong to the Canarian island population, so it's a stable population, as I showed in the yesterday presentation. Here you have a quick overview of the traits. Because many traits were measured on the animals, were recorded, they had to be somehow aggregated. So you have for zoometrics, lengths, heights, widths, and perimeters, in total 30 measurements per animal. We had also for biomechanics, acceleration, velocity, displacement, and spatial position. And for behavior, basically coping styles, general cognition, intelligence, and intelligence quotient index. So if you are interested in more details, I will go very quickly. I will not go through these details, but you can find publications mentioned here where you have all the methods also explain how to collect these data, how to analyze the 2 video data, and how to get the behavioral traits measured. So, anticipating something I will be talking a little bit later on in the next session, this study has been done using a SNP array. A SNP array is a tool that allows you to genotype and thousands hundreds of SNPs of genetic markers. You can see here the number of markers that were used for this study, so covering the entire genome, and basically showing very quickly the results. When we consider the genome level significance, which is the one more strict, we got an overall 16 loci significant for action of selection on these loci. You have Manhattan plots and cuckoo plots for some of these parameters that were considered in the study. But the most important information comes from this slide, where you see the genes that actually are located close to the markers that were identified as significant. So these genes have been categorized based on they are shown based on the traits they were significant for. And you see that there are some of them that are with color codes. It means that they have been detected in different traits, but normally within the same trait category. So for behavior, different traits, but all within behavior, within zoometrics, or within biomechanics. And that's not surprising, because we know that some traits within the same category can be correlated. So we can have like a pleiotropic effect for the different genes. This is quite relevant because it's a sort of first list of genes for some of these traits that have never been considered before in similar genome-wide association studies. So it's a very preliminary study, but although it has been recently published, so you have the reference here. And also just to mention that in addition to the publication, we also trying to dig more into these results. And when we do some gene network or gene ontology analysis and looking at the biological processes that the genes we found relate with, basically you can find with different approaches, we come out with similar results on response to stress, nervous system process, locomotion, sensory perception, neural system, I can go quickly, postsynaptic neurotransmitter receptor activity. So in any case, we get gene ontology terms, molecular function that are strictly related with the phenotypes we were interested in working with. So it means that although this approach is very prone to false positives, most of the genes that we detected are probably really selected genes, and so they are candidate genes for further use in selection in dromedaries. So basically, this is the conclusion I'm giving you already. This study is very preliminary, but the information we got, once validated, because validation using independent set of animals is fundamental after this first stage. Once validated, they can be used for assisting breeding programs. But what I want to stress here, that the considered traits also point to the need of considering health and welfare in genetic schemes for these animals. And that's all. I think I was on time.
We have a lot of time.
Okay.
We have actually half the time.
Thank you, Elena, for your presentation. I have seen that U6 is the only gene present in the 3 categories. So how about this gene? So what is the function? Just give me some insight about that.
Actually, I'm not the one who did the analysis, so I cannot answer to your question. But I'm not surprised anyway, because even when we did, we have been working a lot, not with camels because we had no phenotypes, but a lot with sheep and cattle and goats as well. And it happens that sometimes you have very basic molecular processes, so you can have the same target genes that in one district do something and it do completely different things in another district. So we will be exploring more. But what is most interesting is try to validate these signals. That's the next step.
Hello. Very interesting. I want to know, how do you measure intelligence in a dromedary?
That's a good question. I don't know if we can put back the presentation, because there is a number of operator-based ways. So for sure, there is a standardization that you need for this phenotype.
Yes, but
Yes,
I
sorry.
want you put the animals in a trial and in an experimental situation
Yeah.
to solve the problems.
Yeah, exactly. So these
okay.
animals, I forget mentioning that they are from Canary Island and they are used for leisure, for tourism. So basically what they did, they create new situations to the animals and they see how they can react. Either how they react in terms of fear, in terms of coping styles, or how they can solve problems. So if I put a problem in front of the animal, how they can, for sure there is some, let's say, subjectiveness in the way you measure this trait. And this is one topic where for sure we need increased standardization. But I know that they, the group who did these phenotypic measurements is from University of Cordoba, and they use a protocol that has been previously validated on horses and donkeys. Sure, they are different animals, and that's why they also had to adapt to the dromedaries.
Okay, thank you very much.
You're welcome.
One more question. Anyone behind? Okay, if no questions, thank you very much. Title for the next presentation is Genetic Diversity Analysis of the lactoferrin whey protein coding gene in New and Old World camelids. So how to use this one? You need to hold up that.
Okay. Well, today I'm going to present instead of Nerose Letayev because she's not here. She was supposed to be here, but she experienced some problem in visa not to come here in Morocco, but she got expired her visa in Italy. She's from Tunisia, and yeah. She's still in the process to get new one, so we decided that it was not the case to come here, otherwise she could get problem to get inside in Italy again. So very quickly, oh, what's happened? Okay, this is the outline of my presentation. Why did we choose lactoferrin gene? Which is the main objective? How did we proceed? Which result we get? And then I will move to the conclusion. Very quickly, of course, we know that milk is a very complex matter, but if we look at the protein side, we can separate the casein from the whey proteins, and the ratio between casein and whey proteins are important for many aspects. I will focus my attention on lactoferrin, which is a component of whey proteins together with the other whey proteins like alpha-lactoalbumin, immunoglobulin, lactoferrin, and so on. Lactoferrin is expressed in mammary gland during the pregnancy. They have several functions. One of the most important functions is to prevent the fat goblet aggregation during the secretion in the milk. And in addition to this, one function is to prevent the occurrence of infection, and so it's a certain protection role against the mastitis in lactating animals. Its concentration in the camel milk is about 3 times more than what we can find in the cow milk. So we are talking about 950 milligram per liter compared to 300 milligrams per liter of milk in cow. So far, the information present in the website tell us about 2 non-allelic variants which are described in the literature. So those are the result of alternative splicing, but there are no information about alleles so far present in the literature. So let's say that the objective of this work, which is actually preliminary, so we still are going on to analyze the function of this of our result, was to investigate the promoter and the coding region of the gene in all the new world camelids to discover genetic diversity that we could link to a potential function of the protein. What we did is just to collect the samples, to resequence the gene. Of course, the information is already present in the genome, but we resequence with the idea to discover genetic diversity. And so when we found genetic diversity, we went to the bioinformatic analysis and then finally genotyping. At this step, we stopped for this preliminary work. So what we did is to confirm, of course, the gene structure, which is actually a small one. It's a 2,272 base pair, which is divided in 4 exons and 3 introns. And actually, the protein is not that big. In fact, it's only 154 amino acids. And the stop codon is just in the last exon. In addition to this, we sequenced and analyzed the biodiversity found in the flanking region, just upstream, not that much, about 650 base pair upstream the first exon, and also about 300 base pair downstream. This is the regulatory elements are there to indicate how much the gene can be expressed or suppressed according to the role. This is an overview of the genetic diversity that we have found. So, of course, dromedar, Bactrian, llamas, and alpacas. In dromedars, we have found 14 polymorphic sites. Actually, we were looking for mutation in the exon, which could bring to amino acid change, but in dromedars, we could not find it. Then we analyzed the Bactrian, which resulted the less polymorphic from this point of view. And for us, it was a little bit surprising because normally in Bactrian, we found more mutation than what we can find in dromedars. And even in this case, we could not find mutations in exonic region. The most polymorphic were the South American camelids, as normally happens. In llama, we found 38 polymorphic sites and 2 mutations detected in the exon were responsible for amino acid change. And a similar situation also in alpaca, where 31 polymorphic sites were found, and 3 out of 5 mutations found in exons were non-synonymous mutations, so bring to amino acid change. We investigate the gene promoter to establish if any of the SNP found in the promoter could affect actually the transcription factors which activate the transcription of the gene. We found some interesting stuff. For example, in dromedars, we found one mutation which actually brings to the in the nucleotide, which is indicated here. It's a polymorphic one. Of course, the analysis has been done with homozygous animals, and this variant brings to the activation of one GATA transcription factor, which could activate more the expression of the gene. In llama, we have found 4 mutations in the promoter, which more or less all bring to activation or removal of a binding site for transcription factors. And from this point of view, we still have to investigate the role of all of them. And alpaca, more or less the same. So we found 3 SNPs in the promoter which could affect the gene expression, creating or removal transcription factors. So the analysis is still going on from this point of view. What we did in addition is, after finding the mutation, to confirm it at the population level. So the first step actually is to see how spread are the mutation in the population. So we found interesting SNPs in both alpacas and llamas. We bring this brought out to the idea that probably this SNP has been already present before the diversification of the 2 species, or maybe when they mate each other and then separated. We don't know, so we still have to understand that. By the way, in the promoter region, we focus the idea on one SNP, which is responsible for the creation of this transcription site, MBP1, and the one mutation in exon 3, which is responsible for the amino acid change glue to lysine. We did our preliminary genotyping. In this case, we proceed by sequencing. It's a small gene, so we can make a Sanger sequencing. we found the very low frequency of the G allele, but actually the distribution is quite clear. So we have homozygous, but we also have heterozygous samples. Instead, for the exon 3, we found we established an allele-specific PCR to genotype faster all the animals. In this case, we reached almost 130 animals to establish the allele frequency. Then we found additional SNP more interesting from alpacas, which were carrying the amino acid change. In this case, we proceed again with the Sanger sequencing. We are still genotyping the animals. So I'm sorry, but this is just a preliminary study. And in conclusion, I can say that it's very promising, the work, because the role of lactoferrin in protecting the animals against the mastitis is it's well documented in the literature. So in addition to this, the activity in the emulsification and the growth promoting of bifidobacterial can be enhanced by the genotype dependent of the lactoferrin. So this is what will be the functional study we will do in the next step. So we will go to investigate also the potential effects of the mutation on the splicing and expression level, taking into account the mutation that we have found in the promoter region. Finally, we will focus on our functional study, mainly in vitro, as a preliminary step before arriving to a final conclusion. So I would like to thank all of you for the attention. I'm sorry for Neiros, which could be better than me at this point because it's her work. So please, if you need further information, write directly to her, which is the best to answer to your question. Thank you very much.
It's a very interesting subject, and you presented well. And genomics and gene typing is very new, modern area in this camelid research. So I like to have many questions possible because we have a lot of time left.
Okay. No, it's just a very general question because I mean, from your work on not only on this gene but on other genes, I was just wondering if you had the feeling that actually you consistently find lower diversity in camels than in New World, and if you have any idea about that.
Yeah, it's complicated to answer to this question. Let's say that we all know from the literature what happened in the dromedaries during their evolution. So it's a result of the bottleneck that actually we experience in our work. So next to you, there is sorry, not. No, not in the South American camelids. That's why actually we experience more diversity in South American camelids than what we can find in the old world, so-called old world dromedaries. I was surprised for this particular gene that the Bactrian camels is less polymorphic than what we can find in dromedaries. But on the other side, the number of animals that I have analyzed from Bactrian is much lower than what I could do in dromedaries. So this could be one reason for that. But generally, we try to keep the number of resequencing similar. So we are not making 3 animals and 45. So, for example, so we try to keep the same number of animals in general.
Okay, thank you very much, Alfredo. My question is, what is the role of lactoferrin in the mastitis prevention? And would it be possible to use this gene as a marker for selection for mastitis, against mastitis?
So there are all the proteins in the whey protein fractions have an effect. Actually, some of them are more, some others are less. For example, immunoglobulin, of course, have the higher impact on it. So in the end, lactoferrin, as many other whey proteins, when they are digested, they produce bioactive peptides, which could negatively, positively impact on the bacteria which causes normally mastitis. So this is the positive effect that they can have. So the link between the lactoferrin and the mastitis is connected to the activity, bioactivity of the protein generally. If can be used or not as a biomarker, I think a pool of proteins as always happens, need to be considered for the positive effects in preventing mastitis.
Any young scientists, local audiences, please, any questions?
Thank you very much, Alfredo, for the nice presentation. I have just one question. Did you get the opportunity to make phylogeny with this mutation just to see how the mutation were inherited horizontally or
Not yet.
No, not yet.
Not yet, because those are results just obtained before coming. So we decided to include also additional slide because they were just arrived. But next step, first of all, is to see the haplotypes, because we have a SNP enough, and we were also surprised because the gene is very small, it's 2,200 base pair to have so many mutations. So the first step is to see the haplotypes. According to the haplotype, we can explore the difference and how this could be related to the differentiation of the animals.
Thank you.
You're welcome.
Thank you very much. And we have next paper lined up. The title is called The Biochemical Properties of some Metaproteins from Camel, presented by Dr. Should I
We have this presentation.
Oh, sorry. Okay, actually I is skipping one. The presented by Dr. Biodiversity. The title is the demographic parameters and factor affecting the development of camel herds in the southern region of Morocco. This sounds right?
Yeah.
Correct. Thank you.
So good afternoon, everyone. So I will be speaking about the demographic parameters and factor affecting the development of camel herds in the southern region of the country. So camelids, with the population of 35.5 million, contribute minimally to the production of milk and meat. It's less than 5% of the global production, either of meat or milk. Camels, although they have a very high quality natural product, and they produce this in challenging climates which other species could not do. So, but they are considered as second-level species. But lately, a spark of renewed interest in this species emerged, mainly for this question, for the challenging climates, because camels could deal with harsh conditions. Historically, camels were not used for production. It was used for trade between these regions, but it shifted with the modern socioeconomic and environmental changes, and now it's used for the production and for the commercialization of camel products. So camel breeding focus on milk and meat production, mainly in remote areas when food supply chains are limited. So despite the high quality products, and despite the adaptability to harsh environment, as I said before, they are they are considered as second-level species. And there is a limited demographic data compared to other livestock species, like cattle or even goats or sheep. And the reproductive performance, it has a good impact on milk and meat in production. And getting reproductive performance is very hard, mainly in extensive farming systems. And for this, there is a big need for methods to assist the demographic parameters. And this is the main objective of our work. We did small work here to try to get the demographic parameters of this species in the southern region of the country. So in developing countries, it's very hard to get data, general data, either production data or genetic data for highly mobile and dispersed herds like camels. And it's difficult to make an individual collection of data too, because it's very costly. And it's hard too, like in other species like cattle and sheep and goat, to make a simplified monitoring systems, above all in nomadic system. It's really hard. For all these reasons, there is a big need to have a simplified system. And here we are suggesting retrospective methods, which is a method in which we track reproductive and demographic events over the last 12 months of the females. And we use this information with statistical methods to evaluate the demographic and the demographic parameters of our herds. So here's the material method. So the studied area was in the province of Laayoune and Smara, which are part of Laayoune-Saqi al-Hamra region. We sampled 31 camel herds using stratified probabilistic sampling. And the study period was in the year 2022, over 1 year, or exactly over 4 months during this year. So the data were collected at water point and at the areas where the animal moves at the, of different parts of the desert, and either during the water point or during the herd movement at the two provinces of Laayoune and Smara. So what we collected is a key indicator like birth rate, mortality rate, reproduction performance. And in plus of this, rates related to reproduction performance, we collected data about the feeding, the health practices, and the labor use. All this data were entered into the Access database with the unique codification of each questionnaire. The redundant or irrelevant data were removed, and the continuous variable were converted into categorical variable to make it easy for statistical analysis. And statistical analysis was done according to the 12MO method to assess the demographic parameters of our herds. So what is the 12MO method? Just in a short way, the 12MO method is a way that use some parameters and some survey of the herds, mainly of the females, over the past 12 months, and use this information to estimate the demographic parameters. So this information concerns the abortion rate, parturition rate, prolificacy, mortality rate, offtake rate, and intake rate. So this hazardous and instantaneous events were recorded on our herds, and what we recorded exactly, and what was very important for our study, is the abortion rate, which was the number of abortion per female, parturition rate, which is the frequency of female giving birth, the birth, either the live birth or the stillborn, the prolificity, which is the number, in this case is 1, and the mortality rate, which is the number of animals that die from natural reasons, and also of the offtake rate, which corresponds to the slaughter and to the sales, and the intake rate, which corresponds to the loans, what we bring into the herd. After gathering all this information, we apply some statistical tools to estimate the demographic parameters. So here a little description. So this is the number of herd that we used according to the size. The size of the herd that we use in our study is between 20 and 70 generally. So here is the proportion of animals according to age and sex. We see that the age of female, it was between, it's between mainly 15 and 20. However, the age of male, it was evenly distributed between 0 and 20. This is because of the difference of number of male and females in the herd, male being at maximum 2 in a particular herd. So this is the parturition rate. It's not mentioned here, but the parturition rate in our study, it was 0.46. And we can see that it increased according to the age of the female. For example, from the age 1 to the age 10, it increased. After that, we have slight decrease till the age of 17. And after, we have, again, an increase of the parturition rate. We would like to model the parturition rate according to the age. And for that, we use a 2 model. The first model is with intercept, this one. And the second model is without intercept. So this is the question of the first model, is parturition rate is 0.41 age minus 1.1. And parturition rate in the second model equal to 0.46 per age. And of course, what this coefficient here is the parturition, the average parturition, additional parturition that we can gain when we have 1 year of increase of the age of female. So statistically, what is this number? This number is the average parturition rate, which is 0.46. We choose it 0.46 rather than 0.41 because there is an information that I didn't report here. The R square, which is the determination coefficient that determines the accuracy of the model, is higher here, is 80% here, whereas here is just 64%. So the death rate in our study was 10%, and there is a difference between male and female, but just because of time, I will skip this. So just what we can have from this study is that the high mortality rate, particularly among males, suggests that there is a place for more improvement. And this improvement could be done by better veterinary care and feeding strategy during critical periods. In this study, we got a higher reproductive rate, which is good comparatively to other studies, but unfortunately, this was tempered by a high abortion and high mortality rate. This year has one inconvenience because it was done over 1 year, and of course it gives snapshot of herd dynamics, but may not capture all the impact because we know that situation fluctuates between each year and the other. So a similar study of 2 or 3 years would be better than that. So as conclusions, camels are well adapted to the southern region of the country, but there are some serious constraints that we cited in this work. But a key takeaway from this is that we need to focus on reproduction parameter and the mortality rate in this part of the country, because those are the parameters that we found that are critical comparatively to the other demographic parameters. And knowing the complexity of such parameter, we suggest that this intervention could be based on participatory approach involving agricultural development services, professional organization, research, education sector to meet a demand for camel products. And thank you for your attention.
Thank you. Thank you very much. Very interesting talk. If you have any questions just after the presentation, talk to him. Ne? So don't have time. Thank you. Sorry. As now we come back to that the next topic with the biochemical properties of some metapholo proteins from camel. 5 authors listed. I don't know who is actually coming up to present that. No, it's actually from here. See, this 5 people.
Abdel Khalil Samad, this guy.
No.
No? Are you the one? Good to see you.
You just arrived?
So, by Dave, can change.
One after here, something. No, who's
We'll give them enough time.
he here or not?
I guess Hani.
Call out. Could you call out?
Yeah, Ba'auda. I don't know how to pronounce it.
Not sure.
Ba'auda. Hani.
Could you call out?
If you are not here, we will invite again. You have to present it. Yeah, so, but here I think Shani would like to.
Yeah, could you actually tell them? I'm the chairman.
I introduce you. Yes, of course. Yes.
Tell them forward.
Pour, ouais, je vais l'annoncer.
Ok.
I will give this presentation. You have to wait.
No problem. I stay here now.
Excusez-moi de ce petit retard qui, malgré moi, donc je croyais que c'est encore le temps. Excusez-moi, to my dear camelids. Donc, notre contribution sur les particularités biochimiques ou bien les connaissances biochimiques chez le dromadaire ont été faites en 2 grandes étapes. Une étape, alors, comment... Ouais, pas. Les premiers travaux ont été réalisés, sur le métabolisme de certains oligo-éléments, à savoir le cuivre, le zinc et le sélénium, et ont été conduits au Maroc avec une équipe Bengoumi, Samadi et Faye, et sur lesquels nous avons abouti à certains résultats. Les travaux ont montré sur l'ensemble que le dromadaire régule toujours ses concentrations plasmatiques en cuivre, zinc et sélénium à des niveaux beaucoup plus très bas que les autres ruminants, comme la vache ou bien les autres petits ruminants. Malgré les concentrations faibles plasmatiques du cuivre, zinc et sélénium, aucun signe clinique ni de déficience n'a été observé chez les animaux de l'expérimentation. Et alors que l'activité des enzymes qui sont liées à ces oligoéléments, à savoir la céréaloplasmine, la SOD, la superoxydismutase et la glutathion peroxydase, restent maintenues et non affectées. Notre hypothèse était, est-ce que la structure et la fonction de ces enzymes seraient différentes chez le dromadaire? C'était la deuxième étape des travaux de purification et de caractérisation de toutes les enzymes qui ont été étudiées. À peu près une dizaine de protéines qui ont été purifiées. Et c'était une équipe composée de Shafiq, Samadi et Mavi. Alors, toutes ces protéines ont été purifiées par les méthodes biochimiques, à savoir des échangeurs d'ions, des chromatographies d'exclusion et une caractérisation par les électrophorèses et les spectroscopies. Alors, sur l'ensemble des résultats obtenus, la première protéine qui a été purifiée, c'est la céréaloplasmine, qui est une enzyme ou bien une protéine cuivre dépendante. La particularité de cette protéine, c'est qu'elle est beaucoup plus acide que celle qui a été observée chez la vache ou bien chez l'homme ou bien chez le mouton, alors que les autres paramètres restent les mêmes. Et c'était la première protéine qui a été purifiée. La purification de la superoxydismutase à partir du foie, du rein et de certains organes a montré que cette protéine possède une masse moléculaire beaucoup plus faible que celle observée, que ce soit chez la vache ou bien chez l'homme, avec une masse moléculaire de 28 kilodaltons. C'est une protéine, pour la première fois qu'on retrouve, c'est une protéine monomérique et non pas une protéine dimérique, cuivre-zinc dépendante, comme on trouve chez les autres mammifères. Elle possède une activité à une température beaucoup plus élevée, de l'équivalent de 43 degrés, et la protéine est toujours active. Et suite également, donc ce ne sont pas des conclusions, mais tout simplement, puisque cet animal, qui est le dromadaire, qui régule ses concentrations plasmatiques en cuivre et zinc à des taux beaucoup plus faibles, on a retrouvé que cette protéine-là, la concentration en cuivre-zinc reste faible par rapport à celle observée, que ce soit chez la vache ou bien chez l'homme. La catalase a été également purifiée et je signale que les enzymes sur lesquelles on a travaillé, ce sont des enzymes qui sont liées au stress. Et donc suite à l'adaptation de notre animal aux conditions désertiques et au stress. De la même manière, la catalase, elle possède des propriétés uniques comparées à celles des autres mammifères, avec une masse moléculaire beaucoup plus élevée que celle observée chez les autres ruminants, ou bien chez l'homme, avec une température d'activité beaucoup plus élevée, de 45 degrés Celsius, et elle a une grande affinité au peroxyde d'hydrogène qui est toxique. La troisième enzyme ou bien la quatrième enzyme qui est la glutathion peroxydase qui est une sélénoprotéine. Chez les mammifères, chez l'homme ou bien chez la vache, elle possède 6 atomes de sélénium. À peu près, on a trouvé le ratio, il est à 50% chez le dromadaire, avec une différence moléculaire et une comment dirais-je, génétiquement, les enzymes de 53 et les enzymes de 21 kilodaltons, et elle est hétérodimérique. Donc, il y a plusieurs unités dans la protéine, avec une activité optimale qui dépasse les 47 degrés Celsius. elle possède un niveau de concentration de sélénium beaucoup plus bas, il est à la moitié à 50%, comme il est à 3 atomes de sélénium, avec une grande affinité catalytique pour le peroxyde d'hydrogène et une grande affinité pour le peroxyde d'hydrogène avec des concentrations en glutathion qui est très faible. L'anhydrase carbonique également a fait l'objet de purification jusqu'à l'homogénéité avec une faible masse moléculaire ne dépassant pas les 25 kilodaltons, avec 45 degrés Celsius avec une activité optimale, avec une activité à un pH beaucoup plus basique vers les 9, et le cofacteur lié à cette protéine, c'est le fer, à la différence, ce qui a été observé chez la vache et chez l'homme. Avec une faible inhibition par le magnésium et le calcium, ce qui lui contribue une séquestration de CO2 par le magnésium et les sulfates, les carbonates de calcium. Une conclusion sur l'ensemble des travaux, ces enzymes, c'est que toutes les caractéristiques biochimiques de ces enzymes montrent la particularité de l'adaptation probable de ces protéines à l'échelle moléculaire, en l'occurrence de ces enzymes et de ces protéines. Et ces protéines isolées chez le dromadaire représentent une particularité unique à l'adaptation au stress hydrique. Et probablement, on est sur le point de mettre d'être que ces enzymes peuvent être candidates ou bien candidats pour les applications industrielles dans l'avenir. Finalement, tous mes remerciements au Camelid Mens et la contribution dans cette intervention avec les amis Shafiq, Safin Nour et Bouchra Ami et ma vie. Je vous remercie infiniment, cher Camelid Mens.
Present.
Any questions? You mentioned in the last slide about camel enzymes for harsh industrial applications. What does it mean? Can you elaborate on that?
No, not yet.
What?
Not yet.
Because what are you looking for?
For des inhibiteurs of enzymatiques, les inhibiteurs enzymatiques de ces enzymes-là.
Me no English.
No.
It's all right. Any questions? Shahada? No? Any questions here? Thank you very much.
Thank you.
Thank you.
Merci.
Thank you. We have the last speaker. Last speaker for this session is not here. Okay, that's good. A bit of rest. Yeah, no more speakers for this session? Okay, if no more speakers for this session, we close this session and we go for the coffee break. No? No coffee break? Why the suffering, my friend? So, okay, we have another session with the keynote speakers, and starting this session with charming Alina. Do come in, Alina. I think you know Alina by now, And Alina Siani is going to present her keynote speech. Please.
Can I use this one? Thank you. As short as possible. A little bit longer than the first one. If this, okay, thank you. Yes. Okay, in the meantime, I can start without the slides, so we keep time. So the presentation will be a quick overview of genomic advancement. So I wanted to start with the idea, what is genomics compared to genetics? When we talk about genomics, the basic idea is that we don't want to look at single or few genes. We are really interested to the overall picture. So we want to study the whole genome. But you know, studying a whole genome is a little bit challenging. It was even more challenging than it is now when the genomics era started. So we had to find some solution. And the best solution we can find, it was like use genetic markers as a proxy of the whole genome. So put pins in our maps, in our genome, like in our geographical map. And as you can see here, depending on the number and the density of the markers we use, we can get a more refined or a lower resolution picture representation of the genome. So at the beginning of this genomics era, we had sparse genetic markers. And also these kind of markers had a number of problems in the reproducibility of the analysis. And hopefully, I mean, likely at a certain moment in about 15 years ago, there was a, we can say, breakthrough technologies that was developed, which is the SNP array or SNP chip. And this technology actually really started what is called now the agrigenomic revolution. So it allowed a fast, cheap, and reproducible genotyping of animals at 10,000, 100,000 of loci in the genome. So we can get a very precise representation of the genome without sequencing the whole genome. So what SNP arrays are useful for? They have been a tremendous impact on a really huge number of applications in livestock, breeding, and not only. You can see here the list: assessing genetic diversity, breed assignment test related with food trackability and product certification, pedigree reconstruction or pedigree verification, paternity testing, inbreeding, monitoring, and so creating assisting mating plans, identify, so understanding the genomic regions that are responsible for traits of interest, either for diagnostic purposes or for genetic prophylaxis and for marker-assisted selection. And as an evolution of marker-assisted selection, also for estimated, genomic estimated breeding value, and then apply what is now called genomic selection. So in animal breeding in particular, the adoption of SNP array technology has really revolutionized the animal genetic evaluation systems and the selection methods. We now say that we can, early in the life of an animal, get the estimation of his genetic value and decide if we want to use or not as a breeders, directly from the information that is contained in his genome. But I want to stress, so for sure this is a big advantage in terms of genetic trends, as you can see here, with greater performance from genomic selection bulls compared to traditional selection schemes. But what I want to stress here that genomic selection, yes, you can get estimated genomic breeding value from only the genotype of an animal, but this doesn't mean that you don't need any more phenotypes. On the contrary, you need phenotypes that are collected in a very standardized and under a routine protocol, at least in the training, what we call the training population, which will be your reference population, which has to be representative of the population you want to select for. So phenotypes are relevant also in genomic selection schemes. In order to fully exploit the potential that is behind SNP arrays, we also need some other basic tools. And one of these is the reference genome sequence assembled at the chromosome level and fully annotated. Here you have an example of the cattle where you can easily go on Genome Viewer, browse the genome and see wherever the genes are located. Genes or gene features. So where we are with camels. Oh, sorry, here. Otherwise I spoil all the information. What about the reference genome? In 2019, there was the first release of a fully annotated genome at the chromosome level. It was released by the group of Pamela Burger from Vet Meduni, Vienna. It was based on an African female animal, and it was named Camdro3. More recently, in April 2024, a new version has been released, so be aware about that, and is based on a male animal from Qatar. This reference genome also has an advantage that since the father and the mother were also sequenced in the Vertebrate Genome Project website, you can also find the haplotypes, the paternal and the maternal haplotypes. So this can be useful for some specific approaches you want to perform. Both of them are browsable at the website you can find there. So coming to the SNP array, what about SNP array in dromedaries? We have a number of SNP array right now. So we were complaining in the last conferences that we don't have arrays for the dromedaries, but now we have a number of them. And probably the number and the one that I'm showing here is not complete, also because now basically everybody can have its own customized SNP array. Takes a little bit of money to do that, but still it's feasible. So I'm going to just give you a very quick overview of the main, at least the main one that I'm aware about. And as you see, they have been using different platforms, either the Affymetrix one or the Illumina one. So the first one I'm going to present you is the one that has been originated as an initiative by the FAO and the Atomic Energy Agency in Vienna with the collaboration of Vet Meduni group in Vienna. The interesting things in this array is that it's a multi-species array. So here you can contemporarily analyze dromedary, Bactrian, and wild camels, as well as South American camelids. For each of those, you have 60K loci. And in addition, this SNP has been conceived also for use in specific studies analyzing the immune genes. So it has been enriched of 11K immune response genes, SNPs. The SNP design discovery, when you use a SNP array, please always refer to the SNP design, because this is something that can tell you a lot on how the SNP array has been built, and so how it can perform on your specific population. In this case, it was based on 25 genomes for the camels family, so for the is not here the picture, but for dromedary, Bactrian, and wild camels, and for about 40-45 South American camelids. So representative of different geographical areas. You have a good distribution of SNP in the dromedary genome. I'm focusing obviously on dromedaries. And you have a good discrimination power among the different genotypes, either in camels as well as in South American camelids. The same platform has been also used and recently published for a more targeted study, a SNP discovery project that has been carried out in India, so using Indian samples, which means that this SNP array is much more useful if you want to make genetic diversity or selection in Indian population, because the polymorphism comes from Indian animals. And again, good distribution of SNPs, also some SNPs, lower number, but also available for Bactrian camels. I'm now coming to the Illumina one, which is actually a collaborative effort coming out from a proposal that I had the chance and the honor to coordinate. Is from the ICC-GIC, the International Camel Consortium for Genetic Improvement and Conservation. It was funded by Illumina through an Agricultural Greater Good Initiative Award. The main collaborators and project submitters together with me were Pamela Burger from Meduni Vienna, Samantha Brooks from University of Florida, and Faisal Almaden from King Faisal University in Saudi Arabia. I want to mention and to openly take this opportunity to thanks also for Morocco Professor Mohamed Piro, who actively participated in the project. And for sure, I cannot avoid mentioning the PhD student, Silvia Bruno, who actually did most of the bioinformatic job that is behind the development of this tool. So just very quickly, to give you an idea, which are the two main strengths of this SNP design process. The coverage, geographical coverage, you have here 192 samples from 19 countries that have been whole genome sequenced, getting a lot of polymorphism out of that. So we basically try to capture as much as possible the diversity that is present in the species. We had to narrow down the number of loci because they had to fit into the format of the array, and just for commercial reason, Illumina decided to have what we call a medium density. But as you see there, we had more than 600 SNPs that pass the quality filter. So we are even now ready to get a high density version, a second improved version of the SNP array. So the final one, the commercial one, has 56,000 SNPs, some traits and characteristics I'm not going to describe in detail, but I also want to emphasize the second strength point, which is a strict, very strict validation process, which has been based on two different approaches. The first round was based on whole genome sequencing animals from different countries, so a different batch, and to see how much the polymorphism were still there. So we can generalize the use of the SNP array in populations that were not considered in the SNP design process, and the performance was very good. And also, we perform a SNP array genotyping of the animals that had been already sequenced, whole genome sequenced, to check for the consistencies between the genotypes coming from the whole genome sequencing and the genotypes coming from the SNP array. And you see the concordance rates is incredibly high. Also, the genome representation is very good. Almost all the chromosome has a very, very good representation. So now it's commercially available. It's still, I say cheap. What does cheap means? It's still a little bit more expensive than the similar tools we do have in sheep, because we need to use it in order for the price to go down. But the interesting point is that in the few months after the commercial release, we already get information from the company that actually more than 12,000 dromedaries have been already genotyped using this tool. So just a very huge amount of animals. How many of them had phenotypes? We don't know, but very few probably. Some of them were also genotyped under the frame of a EU-funded project, Camel Shield, that is just going to the end right now. And I just take 1 minute, but I really want to give the paternity somehow to this long process to Laayoune, because we have been, actually, as I mentioned before, we have been conceiving this project and submitting this project, which means preparing as well the project during the Laayoune conference, the ISOCARD Laayoune conference in 2018. So it took some time. There was pandemic in the middle, everything, but in the end, we do arrive at the end. The same platform has been used also by the Ministry of Agriculture, Water and Environment in Kingdom of Saudi Arabia, this time focusing on Saudi camels. So again, more specifically being used for animals in that country. So just a number of spin-off things that come out, because we said in the beginning, a SNP array that can pave the way. What are the impact of the SNP array and the sequencing project? We now have a large set of fully sequenced whole genomes. What we can do with that? A lot of amazing things. So a lot of room for collaborations at different levels. A number of regional and global diversity studies that have been performed. I'm mentioning here not only the one that have been done so far using SNP arrays, but also the one that have been performed previously using other technologies that still give us a representation of the whole genome, like the genotyping by sequencing and the whole genome sequencing approaches. And I try to divide them either if they were worldwide or if they were regional. And for the regional one, you see India, the Arabian Peninsula, Spain, Algeria, and Morocco in a few minutes. I would also try to answer to the question. You raise a question, Professor Zrairi, today. You asked how and when the genetician will tell us if we have or we don't have breeds. That's an hard question to answer. But in general, most of the studies that have been done, most, not all of them, they had difficulties in finding genetic structuring. So genetic evidence of the presence of breeds. So we are a little bit, I think, in most of the cases, far from calling them as breeds. Then, since we have a lot of whole genome sequences, we can go targeted, and we can have targeted genes analysis, so far, like the myostatin genes at camel immunoglobulins and so on. We open the way for genome-wide selection signature studies. Every time we don't have phenotypes, which is very common in these species, we still can investigate the molecular mechanisms that are behind some general phenotypic traits using this selection signature approach. And you have here a number of studies. And you see from the bottom to the top that the first published one were mainly focused on growth traits because it were a little bit easier to measure. But time by time, we also included some other kind of traits like trypanosomiasy, and I will even show better this point when we talk about genome-wide association studies, with starting from growth rates, morphometric traits, and then also, as I presented before, behavioral related traits, coat color, milk yield, and also health traits. Much more we are expecting to come because we have many traits. We just miss phenotypes. I stress this again. And I also mentioned before the need for validating the signals. I know I'm really close to the end. Validation, what does it mean? If we want to use this information, we have to be sure about our information, and validation is the only way. Up to now, to my knowledge, this case of the group of Professor Hassan Al-Haddad in Kuwait University trying to validate the role of the FGF5 gene in the length of hair in dromedaries is the only, unpublished yet, but is the only tentative validation study. Okay. Parentage, there is some group in Europe offering DNA SNP array-based parentage testing for genealogical proving. Phenotypes. Due to the emphasis on phenotypes, quickly mentioning again some work that has been done, great work that has been done by Hassan group, Hassan Al-Haddad in Kuwait University, to somehow standardize a research methodology for collecting phenotypes, for collecting and archiving biological material, and for using the most current methods for morphometric analysis. You have here just some publications that have been released about using image-based morphometrics, coat color, coat length, texture, and for sure in Gulf countries there is a big emphasis on show camels. We saw today the cost and the value of those animals. So there is a lot of interest on that, with even some specificity for the head, the shape, and also want to mention the work that has been done by University of Cordoba that is actually part of the presentation I gave you before, in addition to hair fibers analysis also in dromedaries. So where to go? That's the last one. Quickly. We can use the available information to improve the reference genome, but we need collaboration among different groups. We can, again, collaboratively design a high-density SNP array. At this stage, we don't have representation of SNPs in the Y chromosome. We can go toward the pangenome, but there is some challenges. For sure, we need policies for data sharing and meta-analysis because SNP are reproducible and we can share data and make further and powerful analysis. We can dig more into the available sequence to extract further information. We need validation, validation, validation of candidate genes. We need to extend the genotyping effort. We want to cover large fractions of national populations, and for that, for sure, we need some policies at national levels. We need to establish, empower, or validate genealogical book, and for this, we need the role of breeder association. We need standardized routine collection of phenotypes and collection of metadata of the phenotype animals, human capacities, facilities, interaction between the quadruple helix, and strategic, long-term, sustainable vision. And thank you.
Thank you very much, Elena. Any questions? There is, yeah. Wani?
Thank you, Elena. Nice presentation. Some of which goes above our head because we don't know much of these genetics. But the basic question I wanted to ask was, like, do we know the genes now which are responsible for the coat color? Number one.
The coat color.
Coat color in camels. And can we modify those? Can we change them? Like, can we change a black camel into a white camel? Number one. Number second question, do we have the SNPs now for, like in cattle, we can say that this is a good animal for breeding. This is a good animal for milk production. This is a good animal for races. And this is a good animal for other purposes. So do we have such SNPs for camels as well now available?
I'm very sorry to disappoint you, not yet. Because for sure what we need to get this information, as I was saying, is to really get a lot of studies supporting the same information. So we need to get a lot of studies that confirm a certain SNP as causative of a given phenotypes. At this stage, answering to your first question about coat color, there is a number of papers published mentioning specific SNPs that are responsible for white color, for few aspects of the color, because you know that coat color is also a very complex phenotype. The second question, I don't know how to answer. You have the answer. Can we transform using biotechnologies? I would say using selection, because this is our tools. Yes, it's feasible, but it would need times. And also it would need more studies to understand the extra genetic mechanism that can explain also this phenotype. I mean, we have to think that one complex phenotype cannot be just like myostatin, one gene, and almost all the diversity is explained by having or not having the allele. So there is, as someone else previously said, I think Alfredo said, it's not a single gene. It's probably, it will be a set of genes that will be needed to get a specific phenotype selected for. Not sure if I answered your question.
No, thank you. Only the thing, like your group, the FASL from Saudi, they have recently started the project on where they select the camels, the milking camels, on the basis of some SNPs. Now, this is recently I came to know. That's what I was asking. Are there some SNPs where we can select these camels for, these are good camels for milking, and we keep those camels, and otherwise, if they're not good for milking, we just put them for the other uses?
Yeah,
Thank you.
There is, if you have phenotypes, you can go toward this direction. But I want to stress again, before taking any decision in the field and before making errors, you really want to be sure about what your alleles means.
One last question? No? This lecture, this seminar is a great opportunity, opening the doors wide for collaboration. And it's a great opportunity for ASARI researchers to collaborate with Elena and her group to contribute more to the field. Thank you very much for the presentation.
Yeah, because I'm chairing.
You're chairing. But what we're having now, someone whispered that the coffee is ready. So we go have a cup of coffee and back to continue this session. Thank you.
وإننا اليوم هنا نحن ندافع عن الجمل دفاعا مستميت.
Maybe you want to call them to take a seat.
Ladies, gentlemen, please back. Time to start the session, please. Wabid, Badawi, where are you, mate? You here? Yeah. Well, I hand the microphone to Elena to chair this session. Thank you very much.
Thank you, Rafat. Let's start this last session of the day. It will be 3 more keynote speakers. I'm happy to introduce you already know, Professor Bouabid Badawi from ASARI Laayoune. He will be giving a speech about genetic diversity and evolution of Moroccan camels through SNP analysis. Thank you very much. The floor is yours. Just to mention for all something happened. For all the speaking, I will give you 18 minutes plus 2 minutes. 18 minutes plus 2 minutes for the questions. I will be getting up at 15.
Not
With this one. I was using this one.
This one is good.
Yes. Okay, so you can start.
Okay, 1 second. Give them 1 second. Okay. So hello again. So I will be speaking about genetic diversity and evolution of Moroccan camels through SNP analysis. So during this presentation, we will try to answer one question, is about the camel population in the country. We would like to answer the question if we have one camel population, 2 or 3, with different approach, and we hope to have some conclusive conclusion at the end. So let's start with an introduction. So the camels in Morocco is one humped animal with which we name Camelid dromedarius. We have quite 200,000 animals, and over 80% of them are located in the southern and southeastern region of the country. Camels are used mainly for the production of meat and milk, and they contribute modestly to the global production of milk comparatively with other animal livestocks. So we have quite 14,000 tons of milk and 3,600 tons of meat, according to the Ministry of Agriculture for 2017. Camel population in the country are identified by their morphology, and according to the definition of the breeders, we have in the southern region of the country 3 population. It depends in the regions. Some regions they define 2, some others 3, but general picture we have 3 population, which is Marmori, Khawari, and Ghazni. So as I said before, the Moroccan camel classification is primarily based on morphology. And we have scarce genetic studies that try to understand the structure of camel population in the country. Here I'm citing an example of Piro and al. in 2021, in which they studied many camel population in the country, and they have found a little differentiation between the camel populations. But in this study, they used all the camels from one region, and they used microsatellite markers, and the number of microsatellite markers used was just 6. So it was very few. So we can say that it remains a lot of uncertainty about whether camel Moroccan population represent one group or mixed of populations. So here what I'm speaking about. So according to the definition of the breeders, we have 3 populations of camels. We have the Marmori, the Khawari, and the Ghazni. How they are identified? They are identified by the breeders. And according to the breeders, the Marmori population is known by the production of milk. The Ghazni by its ability to produce more meat. And the Khawari, it has an intermediate production between the two, producing at the same time milk and meat, but in lower quantities for both. So generally, over all the world, the structure of the camel population were studied in many countries, and the tools that were used as genetic tools is autosomal microsatellite, mitochondrial DNA, and recently, many researchers use genotyping by sequencing, and and whole genome sequencing. And very recently, as Elena Chani mentioned in her presentation, we have SNP chip, with which we can use to genotype animals. And in this study, I will present a preliminary result of using one of those chips to genotype the camel in the country. So let's remember the objective of this study. So the main objective of the study is the characterization of the camel population in the southern country in the southern parts of the country. We would like to answer the question just to remind you if we have one population or many population in this region. So for that, we adopted many approaches. The first approach is the morphometric analysis, in which we measured many morphology parameters, and we use this information to estimate how many clusters we have in the population. The second approach, we used microsatellite markers, and we use it to estimate the genetic diversity and the relationship between the animals, and at the same time to see how many clusters we have in the population. As a third approach, we combine both morphometric analysis and genetic analysis using microsatellite, hoping to have a more comprehensive analysis and study. And at the end, we used a genomic analysis using 60K SNP chip with collaboration with, I have to thank the group of Badr Ben Jalloun and Al-Khsasi and all the colleagues there that helped us to do this analysis, to get the high resolution genomic study to assess the genetic variation, and hoping to have a conclusive conclusions about the subject. So let's start with material and method. So quickly, for the three first objectives, the morphometric analysis, the genetic analysis using microsatellite, and the combination of both. This is the animal material we used. So we used the three populations, Mamori, Khawari, and Ghazni, for which at the same time we take 14 morphological parameters, and for the same animal, we extracted blood and we extracted DNA. We extracted DNA from the blood, and we used 16 microsatellite DNA markers. So to do two types of analysis. The first one using the morphological data, and second one using genetic data, and as a third approach, as I mentioned before, to combine the both methodologies. I think there's a problem with the battery here. Anyway, so as a material method too, so for the last part of the study is the genetic analysis using 60K SNP chip. So we took 66 Moroccan camels, genotyped using the Axiom camel SNP array. The analysis was done using the Illumina platform for high throughput genotyping. The chip contained roughly more than 62,000 prop set, but after filtering, we kept 45,000 prop sets. So for this analysis, the quality, a rigorous quality control was done for this kind of study. Just I have to give some example. For example, for the missingness of individual and SNP, we removed all SNP and individuals with excessive missing data, so to keep the integrity of the data. For the minor allele frequency, we exclude all the SNPs that have MAF less than 1%. And we removed too all SNPs that have were not in the Hardy-Wimber equilibrium. At the end, many SNPs were removed, but we kept all the 60 samples that passed the call rate with the percentage more than 99%. Ici. Non, mais je pense qu'il y a un problème de batterie, mais bon, on fait que ce sera pas suite. Okay. So here are the analysis that we did with this 60K chip. So the first, we computed many diversity parameters. The first thing we computed is the observed and expected heterozygosity, because we would like to assess how many genetic diversity we have in our population. And later, we made principal component analysis, which is the standard tool to assess how many clusters we have in our population. After that, we studied the linkage disequilibrium analysis, because this is done to estimate the association, how the alleles are associated more than what happens randomly, if there are any alleles that are associated in non-random way. And at the end, we use discriminant analysis of principal component, because you will see the variability explained by PCI in this case is very weak. And to find solution to that, we use another method for clustering, so to explore the genetic structure of the animals. And we use special approach for that. I will speak about that later. So here the result and discussion. Let's start with the morphometry and the microsatellite analysis. Here in the left, you can see the result of the morphometry. And we can see that we have no clustering. Quite all the population are overlapped. So we can say that the morphometry is not able to distinguish between the camel population. And if we make a conclusion using the morphology information, we can say that we have one population in the country. But when using microsatellite, we can see some clustering here. We can see some clustering. Here we have the population specialized in meat, and here in milk, and here the mixture of milk and meat. So the difference between morphology and genetics, say, for example, we didn't find any difference using the morphology because the morphology, it depends on the genetics and at the same time in the environment. And the fact that the animal are managed in the same way, for that they get homogeneous morphology. However, even they have homogeneous morphology, the print or the difference, they are always kept in the genetic using the microsatellite. So here is the same picture, just here we projected the centers of the population, so we can get a clear idea about the distance. This is the simple Euclidean distance between the population, so to see how each one is far from the other. Using the morphology, we can see that each population is near to the other ones with no separation. Using the microsatellite, we have some separation, and we have substantial space between the center of each population. So to go further to this analysis, we try to estimate the membership of each animal according to its population. And we find that the membership for the morphology is 0.82, 0.24, 0.42 for camels of meat, or for Ghazni, Khawari, and Marmori. However, using genetics, we find the same thing for the meat, but we find more probability of membership for the mixture, which is Khawari, and the milk, which is Marmori. What we can deduce from this is that either using morphology or genetics, we have a good probability of membership for meat. This means that animals in the southern, or camels in the southern part of the country, were selected mainly for meat production. For that the genome kept signature of selection for this character. However, for milk, we have quite weak probability of membership, highlighting that the milk was, the camels in the southern part was not selected for milk, or at least was not selected enough to see that as genetic selection in the population. After that, we tried to have what are the genetic markers or the morphologic markers that are responsible for the clustering. Of course, we can use morphology too, but I will concentrate on genetics. So in genetics, we find one marker that is more likely or probably responsible for this differentiation of clustering between the two population camels. And this marker is the microsatellite CVRL. Of course, microsatellites are neutral and they are not responsible for phenotype changes, but they could be linked to other genes responsible for the differences of morphology between this camel population. And this marker, if confirmed in other study, could be used to structure the population in the southern part of the country. So here, using the mental test, we try to figure out if there is a correlation between the morphology and the genetic study, that because we use the same animal for the morphology and for the genetics. So we would like if there is a correlation between the two. And we found that there is no correlation with the p-value of 0.34. Does it mean that the clustering found in the morphology is independent for the clustering found and the genetic? And this is more or less logic because, as I said before, microsatellite markers are neutral and are less likely to be responsible for some changes in the phenotype. So now let's jump to the result of using the 60K SNP. So here I start just with some preliminary results. So here the minor allele frequency in our population, in the SNPs used in our population. So we can see that the MAF ranges from 0 to 0.5 with quite a uniform distribution, more or less, having at the same time some alleles with very few frequency and others with frequency of 0.5. That means that our population have a considerable genetic diversity, which is good for any selection program that could be done in the future. For the observed, the same for the observed heterozygosy, we have a good heterozygosy, which means that we have a good genetic diversity too. For the expected heterozygosy is more than the observed heterozygosy. This may be a proof that it could be linked to many, many things, but the more likely reason is that we have a substructure within our population. I mean, we have not what homogeneous population. We have many homogeneous population. For that we have the expected heterozygosy that is more than the observed heterozygosy. So here are the result of analysis of principal component. So we found that we have quite 3 groups. This group F, E, and the other groups. Just I have to mention that we use these groups randomly, just which we affect at each animal, at each group randomly, because the strategy would like to use it in DAPC. Because if you don't do that with DAPC, they will always ask you for a group, but to overpass that, you can give it random groups and later make your reasoning. So here we have quite 3 population, F, E, and this group, and E is the most distant from the other ones. But the problem with PCA is that the variability explained here is just 3.5% here and 2.8. It means that the big part of the variability, which is more than 90%, is not explained here. For that, we would like to use DRPC to get more information. So DRPC is used here. So I will jump directly to the result. So using DRPC, we got 1, 2, 3, many groups. And I remember those groups were attributed randomly. But if we look at the general picture, we can see that generally we have 3 populations. This population, and this population containing B and C, and this population containing E, F, D, which is more likely corresponds that what we have in the southern part of the country. We have 3 different populations. And we have many markers that are responsible for this clustering among the population. And these markers could be, if confirmed, could be used to have a structure of the population in this part of the country. So I'm in the conclusion. So camel population in the southern part of the country could be distinguished into milk and meat because it recapitulates the history of selection for this permit, mainly for meat, as proved in this study, because we found no proofs for the selection for milk. We have some mainly genetic markers that could be used to distinguish among those populations, either in the study of microsatellite or in the study of the SNP chip. So, and we have seen that genetics is more powerful than morphometry to distinguish between camel population. So either using PCR or DRPC, we have some clusters, and in the two methodologies, we get generally 3 camel population, which corresponds to what the breeders are reporting. And thank you, because it doesn't pass.
Thank you, Professor Badawi, for this very interesting presentation. I hope there will be plenty of questions. We can take 1, 2, depending on how quick they will be. Please.
Thank you, Abid, for this nice presentation and nice analysis. Just I would like to ask you, two questions. And the first one, did you validate, these results for typology with farmers, with herders and many, and what do they think about, this vocation or this, profile, or which is really meat production? Because now, yesterday and today, we heard a lot about milk production, meat production. Perhaps we have a mixture between the two. Thank you.
Yeah, so thank you for your question. All the information about the breeds, yes, validated with the breeder, not validated by, I got it from the breeder because I cannot distinguish myself between them. So all this information I got directly from the breeders. So they know how to differentiate between the 3 populations, either it's Ghazni or Khouari or Momoi. For the vocation, second part of the question, if it's milk or meat or mixture, it's the same. If you ask any breeder here in the region, they know very well that Momoi is good for milk because the breeders that use Momoi, they got more milk than Ghazni, and they know that Ghazni is more suitable for meat production, and Ghazni is quite cross mixture between both of two. So yes, it's an information that we got directly from the breeders.
We have a second.
Merci beaucoup, Badawi, pour cette belle présentation. Je pense que la structuration génétique des populations cameliennes, c'est difficile de vraiment de parler d'une vraiment différenciation raciale bien structurée, parce que tout simplement avec le système d'élevage ouvert, donc il y a tellement un flux de matériel génétique entre les différentes races, donc ce qui rend c'est un peu difficile parce que la reproduction elle est ouverte. Elle est ouverte. Donc, c'est difficile, à mon avis, de parler de véritables races sans qu'on n'arrive pas à contrôler la reproduction. Donc, parce que j'en suis sûr que cette structure que vous en parlez maintenant, lorsque vous répétez dans 2 ans ou 3 ans l'étude, vous allez trouver une autre structure. Parce qu'effectivement, à cause de ce flux du matériel génétique entre les différentes populations qui pâtirent dans le même parcours, qui sont dans la même région. Donc, je pense que pour vraiment pour pouvoir parler de vraies races spécialisées et pour pouvoir sélectionner aussi ces races. Donc, il faut revoir les contrôles de la reproduction. Merci.
Oui, donc oui, je concorde parfaitement qu'on ne peut pas parler de race, ça c'est sûr et certain. Et c'est un snapshot de la situation actuelle, c'est-à-dire qu'est-ce qui se passe actuellement. Je pense que ces études peuvent être intéressantes dans le sens où
You can speak English.
Ah, okay, yeah. So I agree with you, and I think what we have done is snapshot what is happening now. So we have a snapshot of the actual situation. So the actual situation, we have this. And it's more or less, I cannot say it's permanent situation, but it's sustainable situation, because if we have this situation, it's the result of some breeding practices, because our production is managed. It's managed by the breeders, because when they want to keep animals that produce milk, they have generally a herd with one male normally. When at the maximum two, but generally one. And this male, it corresponds to the breed. For example, if they want to produce meat, they bring a male Gazini. And if they want to produce milk, they breed it Marmori. So there is some practices that are maintained and which make this small clustering, relative clustering, sustainable. But I agree with you. For me, this results could be starting point in the case we want to structure the population in the south. And so we have starting point. So here what we have, would we want to have a milk population or milk breed at the long term? Would we want to have a meat population? So this is the situation from which we can start to go ahead.
Thank you so much. Thank you. No, sorry, I think we have to skip to the next speaker. I have been asked to be very strict, sorry. So we move now to the next speaker. We also change a little bit the topic. I give the floor to Professor Foziki Chu. I see you here, yes, who is going to give a presentation about updates on the current situation of camel health in Morocco. Thank you, the floor is yours. You can use that one and point this one over there. Yeah, in this direction and not in that direction. Thank you.
Okay, thank you, Elena. First of all, I would like to thank colleagues from ASARI for inviting me to attend this very interesting Congress. And it is also an opportunity for me to talk about camel pathology in Morocco and the challenges and advances in this field. So can you please help me to how to make this work?
Look if there is fine.
Oh, yes. Okay. This one?
Yes.
Okay.
Back.
This one. Okay. Thanks. Okay. As everybody speaking during this last 2 days, they have been stressing about that camel breeding and production play an important economic and social role in the Saharan and oasis area in the country. However, the sector is constantly threatened by various constraints, including health issues. So the aim of my presentation is to bring insights on the main prevalent and most investigated diseases and pathologies of dromedaries in the country, depict the current situation and methods for their control. And also, from time to time, when it applies, emphasize on research perspective, able to provide more knowledge and answers and possible solution for better health of the national camel herds. To start, I will talk about first about gastrointestinal disorders in the camels. Helminthiasis first are parasitic disease caused by infestation of camels by worms. They have a deleterious impacts on livestock production. There are many examples of several species or genera of parasites that infest camels that has been published worldwide, several countries in Asia, in Middle East, and also in Africa. Treatment of this disease and control are based mainly on the use of anthelmintics. So in Morocco, this infestation has been investigated mainly in the 90s, and many publications have been published. Parasites of the obomasum, of the small and large intestine, including the main devastating parasite like Emanchus contractis. I won't go through much details. I'll just give you, because of the time, we don't have to go to details. So, as I said, most of this publication or investigation took place in the 90s. So, data need to be refreshed, taking into account concern about anthelmintic drug resistance that may be linked to misuses, and also the changes in the environment and climate changes, especially the relations parasite, animal, and environment. This is important. So another problem of gastrointestinal concern is the camel-calf mortality and diarrhea. So an average of 20% camel-calf mortality was found in several Saharan provinces in Morocco. They were mostly attributed to diarrheic syndrome, which contributed to 70% in these mortalities. Barada et al. In 1998 found that 30%, about 13% of dairy calves were infected, were positive to Salmonella, and 11% with E. coli. But still other causes of diarrhea in camels that have not been investigated, which includes other bacteria, Clostridium perfringens, viruses, rotaviruses, coronaviruses, and even protozoa like Cryptosporidium, Eimeria, and Isospora. So camel-calf mortality and diarrhea episodes still to occur in Morocco in several areas of camel rearing in the country. And so there is a need for more and recent investigation and research about all these infections. Research work on viral causes of camel-calf diarrhea in the region of Gelmims and other southern area is being conducted by Dr. Adnas Adnan in the framework of a PhD thesis, and I hope he will discuss and give you a talk and present his preliminary results at the end of this session. So now we go to another group of disease, which are blood protozoal infections. All you know trypanosomiasis, which is known as Surra in the world, and it's worldwide, and it reaches morbidity rate of up to 30% and mortality around 3%. Infection is caused by a blood parasite, which is Trypanosoma evansi, which will lead to severe anemia, which constitutes a real indicator of the disease severity. And we have to stress that the disease is transmitted mechanically by hematophagous flies, such as horseflies and other flies. Camel trypanosomiasis is well known by its vernacular name at the local level as dbab, which is associated by local herders to the presence of the biting fly, dbab, which means in Arabic fly. So a first survey was done in 1988, and the seroprevalence of 18% was observed, and 2 very active foci were detected in the southeastern area in Zagora and Merzouga, with very high prevalence, up to 43%. So treatment with melarsonin is a trypanocidal drug, was found effective as curative and preventive treatment. However, since then, the situation might have been changed for many reasons. First, maybe drug resistance. We have to take that into account. And also modification, the biotope of the vector flies, considering the global climate changes. And an example is we had the flooding in the endemic areas last month. It's a biotope very fruitful for the flies development. So active surveillance and current research data are needed in this field. Another protozoal infection is anaplasma, which has gained more interest, and a result of field survey demonstrated for the first time that about 40% screened camels were PCR positive for anaplasma in the investigated provinces, and sequencing and phylogenetic analysis showed that all strains were 100% identical to Candidatus anaplasma cameli. Now we'll go to skin conditions. I have to say before that although they are caused by different agents, they have common determinants factors for their occurrence and impact. Desert and breeding conditions, which lead to difficulties for implementation of curative and preventive measures. So gathering points, like drinking points, constitute a contamination location or sites between animals and herds. So they do have also a devastating effects on productivity, but also a frustrating psychological impact on camel owners. Clinical signs and lesions are visible to the owners when compared with gastrointestinal parasitism. So we'll start with sarcoptic mange, which is caused by one of combination of several species of mites, mainly Sarcoptes scabii in the dromedaries. And in Morocco, an average prevalence of this infestation was about 27% in the region of Laayoune in 94 and 95. And another investigation found that 12% of the animal were, in 2005, were affected by mange. Another cutaneous disease is the tick infestation. Everybody knows the ticks, because due to desertic environments, camels are very prone to tick infestation and tick-borne diseases. So the most frequently infested ticks of camels are Rhipicephalus, Hyaloma, and Amblyoma. In Morocco, Hyaloma dromedari was found the most prevalent among dromedaries, and the young animals are the most affected, and they get very severe anemia and cutaneous trauma with subsequent dermatitis and abscesses development. So the method for ticks control worldwide is the use of chemical acaricide. Results of a controlled vaccination trial suggested that immunization of camels with the BM86 vaccine, which is a recombinant of Rhipicephalus microplus recombinant, was effective and potential alternative for controlling Hyaloma dromedari ticks infestation in Morocco. So, however, this result still to be assessed under field conditions. Ringworm dermatophytosis, which called by herders in Arabic al-gra'. This is a mycotic skin disease characterized by patchy hair loss throughout the body. The most common isolated is dermatophytes. They have the same trend and impact as mange, but at a lesser extent. Reported prevalence rates were 26% and 16%. Most of affected animals were young, and medical or traditional treatments remain tedious and of relative effectiveness. Now we go to the camel pox. It's a viral infection of the skin in camels due to an orthopoxvirus, which is specific to camels with the zoonotic potential. It's expressed by papules and pustule on the lips and nasal mucosa, but can also generalize to the whole body with most severe extent in young animals 1 to 3 years old. A mortality rate can reach 20%, and also the disease can induce abortions at late pregnancy. About 80% of pregnant infected she-camels will abort. In Morocco, the disease is endemic and called Jidri. Severe episodic outbreaks occurred in southern provinces in 1984, in the summer of 1994. So since then, camel pox seemed to be well controlled by vaccination campaign engaged by the ANSA, because at that time a vaccine was produced by a public pharmaceutical company, and then now there are other vaccines which has been produced and are registered as is shown in the table below. Camel contagious eczema is another viral disease caused by a parapox virus. It has the same changes, but it affects only the lips and the face, and which is induced difficulties of differential diagnosis with camel pox. There were only reports clinically, but there is now a first case report of the disease after confirmation by molecular testing has been recently submitted for publication. So there are other problems with the skin, like caseous lymphadenitis, which are abscesses of the lymph nodes and internal organs such as the liver. And now there is a vaccine available which is included in the same vaccine as the vaccine of pasteurellosis. Another disease is called contagious skin necrosis, which is due to other type or other species of bacteria such as Streptococcus and Corynebacterium pyogenes. So concerning those disease, field surveys need to be investigated, and refreshed situation of these two diseases need to be done. So there are another problem now affecting the pulmonary system, and it's expressed itself as pulmonary syndrome and due mainly to Pasteurella multocida and Mannheimia hemolytica, and it induces fibrinosuppurative bronchopneumonia, very severe in young animals, and may also lead to death of very young calves. Other bacteria are incriminated also in this syndrome, but also viruses such bovine herpes virus, respiratory syncytial virus, and other viruses. So that we don't know if they happen or they occur in camels in Morocco. Yes, in Morocco, the syndrome is called NHAZ, and it's mainly diagnosed on the basis of macroscopic changes. So as I said, this is need for more actual data, real field prevalence and extent of the syndrome and its causes, either bacteria or viruses. 2 minutes. Well, I will finish very fast, I hope. There are endemic infectious disease, very important, I'm sorry. Tuberculosis, everybody knows tuberculosis is very severe disease with pulmonary form. It's caused by Mycobacterium bovis, also tuberculosis. Field prevalence and mycobacterial species involved and risk for public health are not known. So this is an area of research. Paratuberculosis is another mycobacterial disease caused by Mycobacterium avium paratuberculosis. It's chronic enteritis. it has a zoonotic concern since Crohn's disease is linked in humans to this bacteria, and milk from infected animals may be hazardous to health for consumers. Camel abortions, brucellosis is most important abortion cause in camels. Other protozoal infections such as toxoplasma. One thing, it seems also there is a non-infectious cause of abortions that camel herders, some of them are with us here, they know it. It's caused by caterpillar, processionary caterpillars that live on the acacia trees. So when the pregnant camel she eat those caterpillars, they get abortion. So there are other miscellaneous pathologies, edema syndrome, which still not well understood, and may be linked to a chronic enteritis due to Emeria infection in the camels. So we need to answer those questions. We have started the work, but need to be finished. So there are many other viral infections that for which camelid are carriers and potential pathogen carriers, such as MERS-CoV, and also, camel were found positive for blue tongue and West Nile virus, but they were negative to African horses. But this is without showing clinical signs. They were carrier, but no transmission. Also PPR, which is the pest diphtheria minnow, they are carrier, but they don't transmit the disease or the virus. However, Rift Valley fever is an emerging zoonotic viral disease, which happened in ruminants, mainly small ruminants. But in 2010, severe outbreaks of this disease occurred among camels in northern Mauritania. So since then, Morocco has taken some surveillance measures. So the only control of the disease is vaccination. And then a live attenuated vaccine for use in small ruminants has been proven effective, efficient, safe and efficient in camels in Morocco for use in camels. So my conclusion, sorry, just one, 3 conclusions. So several camel health issues are still to be addressed by research, effective and to give effective and practical results and solutions. Need to the participation of professional organization in the process of performing research programs in the field. And last, the involvement of research centers and institutions at local level is crucial for better coverage, effectiveness, and efficiency. And then I take you and thank you for attention. And I'm sorry for
Thank you very much, Professor Kichu. We have time at least for one question. Who want to raise questions to Professor Kichu, please stay here. If we have a question over there.
What a great presentation. Thank you, sir. So you pinpoint the problems for us. You pinpoint or pointed to the problems. I was sitting here waiting for someone to say diseases, and diseases is due management, and the parasites can be controlled, right?
Yeah.
I am not scared of any disease. I'm a vet, like you.
Yeah.
So now, can you give us some recommendation on how to help the breeders to control 1 parasite, 2, whatever you find appropriate? Because these parasites are like destroying the physiology of the animal. And you mentioned to us the abortion and so many things. Thank you so much. Get my question? Should I repeat it in Arabic?
I understood your question.
Okay, sir.
No problem. But I am stuck on the answer.
Then we talk.
It's a big question.
We talk, you and
How
me,
to
and
do
we
it.
put a plan.
Yes.
I will try to help.
It's a big question.
Thank
and
you,
this
sir.
is I think it's a matter of we have to all gather all our efforts and to solve this question. Sorry.
There is another one. We take this one because
Mr.
Thank you for your nice work and nice presentation. I would like to know, have you some results about brucellosis? Do you have results about brucellosis? It's a big problem, and we haven't.
No, brucellosis, the only study has been done in Morocco was done in the 1970s. So there was a group from Toulouse. They made a veterinary doctoral thesis in Toulouse. I got only the abstract. I couldn't reach because it's all documents. I couldn't get. This is the only investigation. They declared they have brucellosis in dromedaries, but they didn't mention what technique they used. Even they isolated or they did only serology. So in this area, we need to do a lot of research because we know one important thing it went not without is a team from the direction of the epidemiology, the Ministry of Health in Morocco. They have done a study here in Laayoune, and they have linked cases of brucellosis in humans with the consumption of unpasteurized camel milk. So this is a big issue. So we know brucellosis is here. And we don't know which is it, Brucella abortus or Brucella militansis. So this is a real area of research because it has a big public health concern. And thank you for this question.
we have a very last question for Professor Kichu.
Thank you very much for your presentation, but you didn't speak about the health of the other others. This is very important point. And in this international symposium or the conference, no found research about this problem. This is a big problem. Did you agree with me?
Yes, I agree with you. There are many other diseases I couldn't go through because of the time, 30 minutes. And they're as important as other diseases I mentioned. Mastitis, metritis, rabies, anthrax, hydatidosis, and many, many of diseases I couldn't go through. These are very important to the camel. So since we are talking about the milk, the others are very important, and mastitis is a big issue for the milk.
Thank you. Thank you, Professor Kichu. Thank you very much. A very last one, okay? Quick one.
This is a herder, a breeder, yeah.
Yeah. A very last one.
Yeah.
بسم الله الرحمن الرحيم. شكرا بروفيسور كاشو على هذا العرض القيم اللي يعالج الصحة الحيوانية. ولا غرابة في أن تتبوأ الإبل وهي سفينة الصحراء وذاكرتها هذا الاهتمام العالمي والدولي، إلا أن لا زالت بالنسبة للأقاليم الجنوبية، الإبل في الأقاليم الجنوبية وبعد التغيرات المناخية تشهد بعض الأمراض الغير مألوفة. في سنة 2005 شهدت الكسابة بعض الأمراض الجديدة وخاصة مرض لورم. هذا مرض لورم يصيب النوق البالغة من العمر 6 سنوات وما فوق. لا يصيب الذكور ولا يصيب صغار الإبل، وهو مرض خطير وليس له إلى حد الآن المختبرات لم تكشف له دواء، وهو يسبب في انتحار كثير من النوق. وفي هذه السنوات الجفاف المتكررة شهدت رؤوس النوق تراجع نفوق الكثير من هذه الرؤوس بسبب هذا المرض. من الناحية الثانية، نحن الكسابة على مستوى الأقاليم الجنوبية نؤكد ونشكركم على هذا بعد المؤتمر اللي جمع بين الباحثين والعلماء وصناع القرار والمربيين، خاصة أن مرض القرع هو مرض وراثي يسببه ذكور الإبل. عندما يكون ذكر الإبل سبق له أن مرض بمرض القرع ويقوم الكساب بإدخاله على القطيع كفحل، هو الذي يسبب انتشار القرع. هذا المرض تخلص منه الكسابة 70% شيئا ما بهذه الفكرة ولله الحمد، لكنه هو طفيليات خارجية، يعني يجب عليك أن تسد عليها الهواء وتقضي عليها بالماء أو بالزيت أو غير ذلك. ولكن الحمد لله المكتب الوطني للسلامة الصحية والقضائية حتى هو انخرط في هذا البرنامج الصحي للتخفيض من هذه الأمراض. وشكرا.
May I translate the question before the answer in Arabic and then I will go through directly? Yeah. شكرا شيخ عبد القادر على السؤال أو الأسئلة ديالك. كيف ما لاحظتي مرض الورم أنا قدمت منه غير واحد النظرة سريعة. هذا متفق معك أنه مرض، وأنا قلتها أنه درسناه أنا بنفسي، كنت اهتميت بهذا النوع ديال الأمراض ودرنا بحوث، ولكن بقينا مازال في إطار الأسئلة لأنه ما تحتش لنا فرصة أخرى باش نزيدوا نكملوا البحث في هذا الميدان هذا. ولهذا أنا تنظن خاصنا نشوفوا كيفاش ننميو البحث في الإبل هذا المرض ولا في أمراض أخرى، ولكن راه هذا خصنا نكونوا نديروا منظومة ديال البحث على الإبل. أنا درت اقتراح بأنه يكون مراكز أو معاهد على المستوى المحلي اللي تهتم بالبحث في الإبل. ما يمكنش أنا ولا منين باحث يجي من الرباط من الدار البيضاء يقلب هنا عليه، خاص نكونوا هنا قراب باش يمكننا نلقاو حلول. السؤال الثاني ديالك ديال القرع، أنه هو مرض انتم تظنوا بأنه وراثي، ولكن أنت عاوتاني قلتي أنه كاين واحد النص منه طرف منه هو طفيلي، وفعلا مرض طفيلي ربما حيت يجي من الاب من الفحل يمكن دونك، ولكن هو طفيلي. وكنشكرك على الاقتراح ديالك، لانه الشيخ عبد القادر انا تقدم لي وتعرفت به بانه هو اختصاصي في الطب في التداوي التقليدي للإبل، وهذا الاقتراح اللي قلتي مهم نستعملوه حتى في الابحاث ديالنا ونطبقوه ونشوفوا واش غيعطي في المختبر، ونشوفوا حتى على المستوى ديال الضيعات واش كاين عنده نتيجه، لانه كيمثل مشكل. انا راه قلتها ما كاينش الدواء ديالو، التداوي ديالو صعيب بزاف. والجمل ماشي بحال بنادم كاين الدواء ديالو. القرع كاين عند بنادم، كاين واحد الدواء لا غريزيوفيلفين كومبريمي خصك تعطيها 30 يوم وانت تعطيها. والجمل شحال خصك تعطيه؟ خصك تعطيه. دونك ما هذاك الشيء اللي كاين عند الانسان ولا عند الكلب ولا القط ما يلقى ما صالحش للجمل. ولهذا خصنا نلقاو شي حاجه اللي هي كتواكب اللي كتنطبق على الابل من الناحيه الفيزيولوجيه، من الناحيه ديال لونفيرونمون، ديال المناخ فين نعيش، البيئه. شكرا. سوري.
Thank you. No, thank you. Thank you again for this rich presentation and rich discussion that will follow also afterwards. So now we give the floor to Professor Pamela Burger from Vedmeduni University in Vienna. She's going to give a talk about camelid research in the One Health context. So please, the floor is yours.
Yeah, thank you. It's working. To the green? This one. Ah, okay. No. No. Okay, good. Okay, now got it. Right. I want to show you our beautiful university. So first of all, thank you very much for inviting me to present today. And Thank you also to all of you to stay until the end of this long day, and I hope I will give you a good entertainment now to the end. And first of all, I really want to ask you to clap your hands and thank all in the name of all of us to this wonderful organization and to the great food and to the great students who are here and help us for the whole organization. So thank you. So I hope you are now awake as well. This is our Vetmed Uni, our beautiful place, and actually it's the third oldest university in the world and the oldest German-speaking university. And it was actually founded for the School of Treatment of Livestock Diseases. It was influenced by the military interest and in the field of human medicine, so for zoonotic diseases basically. And this is our beautiful Research Institute of Wildlife Ecology, and I want to encourage you. I'm looking for collaborations. I'm looking for young students to come with internships, with a scholarship. We have a lot of data to analyze for camels, so if you're interested, please send me an email, and we will find a way that you can come. Okay. Right. So this I'm skipping. If we want to talk about health, sorry, this is very difficult. Okay, about camel research in the One Health context. I will show you something about the One Health principle, about metagenomic and next generation sequencing, and about the importance of the camel immune diversity, immune genetic diversity, also for One Health. We all know about this immense camel biodiversity we have in the world. So how is this now related to One Health? Sorry. One Health is an integrated, unifying approach that aims to sustainably balance and optimize the health of people, animals, and the ecosystem. So we have 3 circles which are intertwined, and I want to emphasize that there is no one circle which is more important than the other. They are all equal. And this is also shown here by the principles of the One Health concept, namely equity between the sectors and the disciplines, parity, multicultural parity and inclusion, stewardship and the responsibility of humans to change behavior and adopt sustainable solutions. And I want to emphasize this again, human behavior to adopt sustainable solutions and the change. Equilibrium for socio-ecological and balance between human, animal and environmental interactions, and transdisciplinary and multi-sectoral collaboration. So this is what we should all aim for. I'm a geneticist, I'm a veterinarian, so I try to find an approach which facilitates and which implements these 3 sectors. And I select One Health genomics, namely real-time genomic next-generation sequencing, which I can retrieve, or which we can retrieve from all the 3 systems, from the animal system, from the human system, and from the ecosystems. So how is this done? We can address with genomic questions, we can research the environmental microbiome, we can do biodiversity monitoring, investigate wildlife crime, screen for zoonotic pathogens, antimicrobial resistance, investigate diversity and productive traits in the different production species, livestock species. And for all of this, we use big data technology and try to combine all these complex systems into one systemic approach. This is a picture of the sequences, of the sequencer and of the sequencing method. It's called nanopore sequencing. And what does this method do? You have a membrane with nanopores, very, very small pores. And the DNA, you just send through these nanopores, and the nanopores, they reflect, they measure change in the current of the flow, of the current flow. And based on this change in the current, you can actually allocate it to the 4 different bases of the DNA. So you can get your complete DNA sequences in one go. We can use this approach also in the field, in field stations, in the tropics, but also in the Alps. So, to put it into a nutshell, the future of your health is in your DNA. And we can use this approach to investigate medical applications, cancer, rare diseases. We can do pangenomes now of the animal and plant breeding. So which means you do not focus on individual genomes, but you investigate altogether a lot of genomes now for the whole species, for the whole breed. So this is done now in bovines. They use multiple genomes between both indicus, so the zebu, between the taurine cattle, the yak, the gaur, and they try to find evolutionary signals, evolutionary genomics between these species. We use it for population conservation genomics and for One Health genomics. So if we put now the camel into the middle of all of this in the One Health genomics, then what can we do with it? We will again retrieve subsystems, information from the 3 subsystems focused on the camel. And with a start, as we started screening the genomes, this is the data also Elena has been showing you at the beginning of the session. We found low genetic differentiation and a demographic bottleneck in the dromedaries worldwide. And also we see that we have differences between the immune genes and the immune genomic variations in the camels. The major histocompatibility complex is one of the most important immune genomic regions for the defense against pathogens. And we see that here the Bactrian camel has the highest variation, and then followed by the dromedary, and then by the wild camel, which is poor in this region. And this demographic contraction or bottleneck, it might have been due that only 6 maternal mitochondrial lineages were selected in the dromedaries during the domestication. But as you can see on this picture, and this is a time axis, this is a time axis from the present here, 1,000 years ago, 10,000 years ago, 100,000 years ago, and 1 million years ago. And you can see that also already before the last glacial period, and here's the glacial maximum, so around 10,000, 20,000 years ago, the dromedary had already a stronger population decline than the both other camel species. So this also accounts for the lower genetic diversity, what we see in the dromedary. We now screened these whole genomes from Algerian and Kenyan dromedaries, and we knew that 2 of these dromedaries were tested negative and positive for trypanosoma evansi. And we used the whole genome data retrieved from blood samples of these individuals to see if we can retrieve these results also within the whole genomes. And we use a specific method for that, and this is a database called the Zuckerberg ID database. And with this you can here investigate metagenomics. You can also search for antimicrobial resistance, and they have a separate pipeline also for SARS-CoV and other viral consensus genomes. So it's a very powerful database. It's still focused a little bit on the human side, but there are a lot of pathogens for animals there as well. So this is quickly the pipeline here. You upload your data. There's a species identification. You can remove the host data and human data, which is very nice. So at the end, you have only sequence reads then in this in your pipeline, which might be due to pathogens or other microorganisms. So we found trypanosoma evansi present in the dromedary, which was tested positive. However, we also found dromedary Trypanosoma brucei positive in the one which was tested negative. So either this is a random result, still we have to confirm it, or the other method used was not so sensitive. But what else did we find? Trypanosoma congolensis, Trypanosoma evansi in some Kenyan dromedaries. And what was quite interesting, we found also different species with Moraxella, for example, in a very high end. Another new, for me it was new, I don't know if some of the vets here have heard about it. It's Stenotrophomonas. It's a bacterial species. It's a complex of bacterial species, and one of them is specifically known as a pathogen, Stenotrophomonas maltophilia, and we found it in a very high abundance in the blood of camel from Algeria. And this is now considered as a newly emerging pathogen of concern, and it's recognized as one of the underestimated important multi-drug resistant organisms in hospitals. So we found this in the dromedary as well. However, searching further for this pathogen in the literature, we see that it's also present in the camel rumen and in the intestinal microbiota, because it is used also to degrade this 2-amino-3-methyl-thionoline. I cannot even pronounce it properly. It's a heterocyclid amine, which is cancerogenic, but camels are able to digest these amines, these heterocyclic amines in their food, because they have also these Staphylomotrophus, Pseudomonas, and other bacteria in their gut. So I just want to emphasize here, there's such a lot of still unknown things we need to understand, and having these methods now with metagenomic sequencing might actually be a good way also to screen, to do genetic monitoring, to do pathogen screening, because with this next generation sequencing method, you are agnostic. You do not a priori search for a specific pathogen, because you are just screening what is there in your sample. And you can use blood samples, fecal samples, swabs from the anus, for example, to see which pathogens are there or which beneficial microorganisms are there. So it's very important, and we can detect very important functions for the camel, for human, and the ecosystem. So this was the part for the One Health, and now I just want to quickly give you an overview about also the evolutionary arms race. It's the interaction between the host and the pathogen. There's always a coevolution between the immune response of the host and the pathogen, which is trying to be even more nasty and to infect even with a higher virulence, for example, or getting resistance to drugs. It's all in this evolutionary arms race. And with this Red Queen hypothesis, it is a hypothesis which was raised that species constantly have to adapt and to evolve to survive. So you always have to run to stay actually at the same place, to stay healthy. You have to evolve, you have to run. I'm skipping the immune system in mammals. T cells, camels have developed a very specific system, somatic hypermutation in the gamma and delta T cells receptors. And I'm just skipping this now, and I'm going to one of our studies, which we did. We wanted to understand the immune response of dromedaries to the Middle Eastern respiratory coronavirus. And as you know, it has a mild course in dromedaries, but 35% mortality rate in humans. And it originally came most likely from the bat, and then has a dromedary as an intermediate host. We investigated 121 dromedaries from 3 sites in the UAE, and we captured 100 immune genes from these individuals and sequenced them, and we found selection signals in the adaptive immune system for MHC1 and MHC2, but also in the innate immune system, which is the first line of defense. And what I personally found most interesting, it also we found selection for this DNA H7 protein, and this is a protein which is actually coding for proteins which help which are a mechanical response. They help the cilia coating the respiratory tract. So your respiratory tract has tiny, tiny cilia which only move in one direction. So when you cough, the cough is not going inside but actually outside, and these are these cilia. And these genes, all of them actually were also associated to respiratory infections in humans and have been found in humans which are infected with SARS, MERS or influenza. And we also found immune response to another zoonotic disease. It's a tick-borne disease. It's the Crimean Congo hemorrhagic fever virus, and has also the dromedary as an intermediate host. And here we found innate immune system genes which were selected. Yes, thanks. Finally, to show you also the highlight of the importance of immune genomic diversity in camels, I want to say that we have these specific nanobodies. Nanobodies can block the receptor binding domain of the SARS-CoV spike glycoprotein against the entry point in the cell, in the human cell. And here you see these nanobodies. Actually, I hope that Professor Muldermann will also give a speech here. I think he was on the list on one of the programs which were sent, so I kept this intentionally short because I thought he would talk about it. But now I'm going to explain it a little bit more detail. What happens here is you have this classical nanobody which has 2 heavy chains and 2 light chains, and these nanobodies, they are a single heavy chain antibody section nanobodies. And what these nanobodies do, they actually can bind to the this site here and block the entry of the virus into the host cell. So with this, if it's working, nope. Yeah, with this, I just want to summarize it and say, putting camels in the middle of One Health, it's also giving us the direction into the sustainability goals, and it is targeting number 3, welfare and good health and well-being of all living people and animals. So camel welfare is also important for animal and human health. Camel zoonotic diseases, the camel immune system, which is supporting also human health. Camels contribute to the sustainable use of terrestrial ecosystems and build resistance to climate change. We have heard from Professor Rafat that they have a lower impact on the soil, and camels are of strong social and cultural importance. That was also mentioned by our colleagues from the New World camels, and many livelihoods depend on it. And with supporting the camelid sector, we can improve the health of humans, of the animal and of the ecosystems. And with this, I wish you excellent achievements in the remaining International Year of the Camelids 2024. Thank you very much.
Thank you, Pamela, for this very interesting perspective talk. We have time for 1 or 2 quick questions, because also I've been informed that we have a change in the program and we have a nice surprise with a new talk to be given, which was not in the program originally.
Thank you, Pamela, for such a nice presentation. I have a quick question about the nanobodies. Recently, I read one publication from US coming from MD Anderson Institute. They produced some mice. They called it nanomouse, which are having nanobodies also. So is it possible to have like camels which produce nanobodies against, for example, any human disease, and which we can use for treatment or drug delivery into humans?
Yes, thank you very much for this question. Yes, it is possible, and they have done this exactly for the SARS-CoV. They infected llamas with SARS virus, and the animals, they produced immunoglobulins, which then help for a passive immunization for the humans. Furthermore, the nanobodies can also be used to act as markers for specific diseases in the brain. So I know that they are now tested for Alzheimer to detect Parkinson or Alzheimer diseases, either one or maybe both. And also they are used to transport a specific medication into the brain to delay the degradation of these, which comes with these diseases. Also, they are used for detecting specific cancers already and to transport drugs into the specific organs for cancer treatment. So they have, these nanobodies have an immense function. And as I said, I saw in one of the previous programs that Muldermans would be here, so there I did not go more into that.
Yes, we have time for another quick question and quick answer.
Yeah, thank you very much. Thank you for your nice talk. So I wanted to ask you something about the Sterotrophomonas you showed. You said that it's a pathogenic species, however used only in metagenomics. So based on what type of genes, 16S, RNA or other genes?
This is DNA. We screened for DNA. Yeah,
16S,
it's not. It's a DNA. It's a bacterium, so it's DNA. And I just could show until now that we have found sequence reads which belong to this family. Yeah. If it's specific, this Maltophila, which is pathogenic.
Just the genus. Not
I don't know yet.
Not the species. Yeah. I know that.
Yeah, exactly.
Most
we have not defined the
most
species.
of the strains are only saprophytic. They are not pathogenic. So maybe you have the species Maltophila, which is, but you can't say this is pathogenic.
No, this strain specifically is pathogenic. That's shown.
But you can't say that the ones you found, thanks to the metagenomics, are pathogenic.
Yes.
This is just maybe you have the genus. You have some representatives of the genus, not the pathogenic ones.
It has the high similarity. So this method, it screens the high similarity of this thing, but I cannot confirm that this is the pathogenic one. Yes. I can't.
This is
just
a part of the 16S.
Yes.
That you sequence.
No. This is the whole sequence.
Okay.
This is the sequence. It's not 16S. It's the sequence reads which are in this database. So it is quite specific. It's already quite specific, this screening.
Yes.
Yeah.
Yes. Okay. Thank you very much.
Thank you.
Okay, thank you. So, thank you, Pamela, again. We have now a presentation that is going to be given by, I hope to pronounce well, Anas Adnan. And the title is Diarrhea in Newborn Camel Calves in Morocco. So, please.
Thank you. 10 minutes. Assalamu alaikum wa rahmatullahi ta'ala wa barakatuh. I will be fast because I am the last one. I am Adnan Anas. I am a veterinarian practitioner in Gulmim since 2014. And now I am a PhD student in EAV Hassan II. So I will go quickly. I will talk about diarrhea in newborn camel calves in Morocco. So this is the outline of my presentation. In the first, we are talking on introduction. Then we're talking the main objective of these studies, material and methods with as a result, and finally discussion. As you know that the newborn camel loss is a big problem, as is the cause of hindrance to breeding development. The causes of this newborn camel losses, we find random herd management, successive dry season, and infectious disease. my optic talking about camel calf diarrhea, we found this symptom with green colors and mixed between green and yellow colors. The main infectious etiology of camel calf diarrhea, we find viruses, bacteria, parasites, and protozoa. The main virus is rotavirus A. Bacteria, we found a lot of Escherichia coli, and the parasites we found gastrointestinal. Protozoa we found Cryptosporidium. Don't forget Emeria, coccidias. So the problematic of this optic, you know that the fruit of breeding is the new camel, born. As you know, the length of camel pregnancy is 12 to 13 months, and the newborn survive is a very big challenge. And this is the problem of infectious disease that. And I sit in Morocco, the most important. The situation of camel calf diarrhea in Morocco and the studies done in Morocco, we have two main studies. The first study is done by Mahan et al. In 1983. They found 25 camel with seropositive to rotavirus. This is a serology study. The second study is done by Bengomi et al. In 1998, and we found 58 diarrheic newborn camel with Escherichia coli, Salmonella, Enterobacter, Proteus, and Cryptosporidium, but not a study about viral causes is doing until now. So the main objective of our study is the identification of the viral and bacterial agent involved in diarrheal disease. We are checking about rotavirus group A and coronavirus, not MERS coronavirus, the coronavirus, bovine diarrhea. Notably that all examined herd are in extensive system, so the follow-up of the herd is difficult, and the restraint of camel calf is very difficult too. The visiting area camel, we visited 9 area camel or 9 herds in Gulmim, Ifni, Tantan, Tiznit, and Shtuka, or Sus Mesa. Material and methods, we noted for each herd location, date, etc. This is the example of survey we are doing. And we take the blood and fecal sample collection from the diarrheic camels. So a total of 40 fecal and blood samples were collected from diarrheic animals. A sterile stall container was used for fecal collection in the interest of conducting virology tests. And serum tube were used for blood samples with the objective of performing serology. And in the last, a rectal swab was taken for future bacteriological analysis. The section of laboratory analysis, we are doing the RTQ PCR. The first step is the extraction of viral RNA. The second one is amplification by RTQ PCR for rotavirus A, B, C, and finally the bovine coronavirus. And we follow the manufacturer's protocols. So I show you the result to go quickly. We examined 203 newborn camel. We found 40 camel with diarrheic syndrome, and the number of deaths are 4. This is the prevalence of neonatal, this is preliminary result, the prevalence of neonatal diarrhea and the mortality rate. The prevalence was 19.7%, and the mortality rate was 10%, based on the result in this table. This is just to show you the different percentage of diarrhea in the different areas. And we found in different areas, we found diarrhea. This is the prevalence of diarrhea according to age of the animals. In our study, we are finding that the aged camel, newborn camel, more than 30 days, have 40% of diarrhea. And according to the sex, we found that the male are more receptive than female, but not a very big difference between it. This is the prevalence according to the status of the mother. Multiparous, 70%, because we have a lot more multiparous than primiparous. Now the most important slide. We found that the positivity rate of rotavirus and coronavirus in Morocco, and this is the first report for these viruses, two viruses. We found 30% of rotavirus group A and 30% of bovine coronavirus. And the co-infection rotavirus coronavirus, it would average weight of 5%. And this is the first of part of this virus. 2 minutes, okay. This is what I'm watching it. The first viral detection of rotavirus A and B and bovine coronavirus in Morocco. For the rotavirus B and C, we are 0% of the newborn camel are not detected. This is just to illustrate that rotavirus A is more important in newborn camel less than 15 days, and bovine coronavirus is more important in camel, newborn camel, more than 15 days. So the discussion, the morbidity and mortality rates we found are lower than those reported by Sani Abdo in 1928, Schwartz and Dioli in 1992, and Abbasi Ali in 1992, but is higher than those reported by Hamid Agab in 2006, Al-Ghoueli et al. In 2012, and Mohammed Ifai in 2016. And this is in Arabi Saudia and in Sudan. The low mortality rate in our study may be due to the treatment given and also the effect of colostrum, because we are treated to animal disease. And the group R rotavirus rate of 30% in dairy camels is higher than those reported by Mohammed et al. In 2003 in Sudan, Khalafallah and Ali in 2006 in Sudan, 20%, Al-Ghoueli et al. 2012 in Saudi Arabia, and Mohammed Fai in 2016 in Arabi Saudia. It's just one. Okay. But lower than the rate found by Hamid El-Jabouri in Iraq, 35% for the male and 37% for the female in Iraq. The last one. The coronavirus rate of 30% is the highest percentage reported in comparison with the study conducted by Mohammed and Fai in 2016, which found a rate of 6% in Pays Arabic Saudi. So the future perspective is to conduct a genetic study to determine the relationship between these coronaviruses and the MERS coronaviruses circulating and causing respiratory issues in Morocco. And thank you for your patience. Thank you.
Thank you very much. Thank you very much, Dr. Adnan. I'm sorry, I guess we are 7 minutes late and I've been asked to be very quick, so we will not have time for questions, but people who have questions, and I'm sure there are, they will approach you and ask directly. I think we have a message for all the participants, and Rafat want to give it, and I declare otherwise close this session.
Thank you very much, Ilana. Thank you. Some housekeeping business, and it's good news. It's about the dinner tonight. So the gala dinner is at 8 o'clock, and 8 o'clock? Yeah, it's not bad. Still got 2 hours, yeah. Buses will pick up those in hotels at after 7:30. 7:30. The bus will wait for 10 minutes. So especially for 2 men, we missed you in the morning. We don't want to miss you for the dinner. And so please, you start receiving people at the venue at 7:30. So if you have the direction, it's at Al-Masira Pivok. Al-Masira Pivok at 7:30, but it will start, the dinner starts at 8 o'clock.
So just to add, for the people that they will come by their cars, they have already received the localization, so it's easy for them to be there. And that's all. Maybe say it in Arabic. دونك بالنسبه للحفل ديال اليوم هاد العشيه دونك غيكون ان شاء الله مع 8. وبالنسبه للناس اللي كاين في لوطيل غيجي البيس ياخذهم مع 7:30 واللي غيجيو بالطوموبيل ديالهم دونك وصفنا لهم لا بوزيسيون في الجروب ولكن كاين يجوا عندي ولا عند الاورغنايزين كوميتي ويعطيهم لا بوزيسيون الا ما وصلتهمش ونشوفكم ان شاء الله مع 8 تما. ثانك يو.
আমি তোরে সব দিয়াছি, আমি তোরে সব দিয়াছি, তুই আমাদের দিলি কি? তোর পিরিতের আগুন বুকে জ্বলে দিকি দিকি রে বন্ধু, তোর পিরিতের আগুন বুকে জ্বলে দিকি দিকি। আমি তোরে সব দিয়াছি, আমি তোরে সব দিয়াছি, তুই